Hey everybody, this is Mr. Later with the last bit of Chapter 18. So this will be one of the longer YouTube lectures. I apologize. This is pretty much the last new material for the year. We condensed some stuff down. Not how I pictured us ending the year, but hey, I really enjoyed all my time that I had with you guys, and I'm going to miss you a lot. So please stay in touch, okay? I'm always here for you, and I love to hear how you guys do after BHS is over for you. Or if you're still here, at BHS next year and the following, please stop by. I love that. It really is the greatest. It's the best to see you guys. So, just another illustration here. You're going to be labeling this with the PIDX1 gene. You can see all the different stuff that goes on with the eukaryotic gene, how the eukaryotic DNA kind of bends and stuff like that. So the transcription factors hit the control elements. There are bending proteins and mediator proteins. And those mediator proteins, they're going to more or less help to attract RNA polymerase, help the DNA to bend, help to bring everything together. And once these transcription factors hit the control elements and the mediator proteins are brought in and the bending proteins, transcription begins. And you're going to label that with the PIDX1 gene. You're going to see that from HHMI this week. Okay, so now we got to talk about a couple of things here. First thing is called combinatorial control, combination control of gene activation. Basically, every single cell in your body has the same DNA, but not every cell in your body reads the same DNA. And that's all due to the fact that your cells have different combinations of control elements. So please read this along. So I'm not going to read this to you, but I tried to make it real, Cliff's Notes version of the textbook. And what this shows you right here, this is the greatest illustration that you're going to see. And what it's trying to show you is this. Both the eye cell and the liver cell have the DNA to produce a protein called albumin. All right, it's called albumin. It's a protein. Only the liver makes the albumin. And here's what they're trying to show you. This is the combination of control elements that a liver cell has. And as you can see, it's got yellow and gray and red. And the liver cell also has the proper activator proteins, you can see their color, which attach to the control elements, and therefore, boom, transcription begins. Now, the liver cell also has the genes to express an eye cell protein called crystallin. However, crystallin has a different combination of control elements. Okay, you can see one is the same here, the gray one, but a different combination of control elements. All right, and the liver cell doesn't have those activator proteins, transcription factors, to read it. If it did, it could, but it naturally would not. Okay, and then here's the eye cell, just the reverse. These are the activator proteins right here that attach to these control elements, the proper combination of control elements, and they attach, and boom, transcription begins. I'm sorry, that would be right here. And that's combinatorial control. And then there's coordinate control. And coordinate control more or less describes the fact that genes in your body have to be turned on at a certain time. If genes are turned on at the wrong time, that's not good. So how is it that certain genes are turned on in sequence, or they're turned on all at the same time, basically? And that's more or less because those genes that need to be turned on in sequence usually have the same combination of control elements and also will be turned on by the same activator proteins. Okay, so that kind of describes that. Now, if you think about like a chromosomal mutation where one piece of chromosome breaks off and attaches to another chromosome, we saw that with the Philadelphia chromosome. That kind of helps you understand why it can cause cancer in that situation, because that piece of gene is getting real close to another gene that might be turned on, and it gets turned on in the wrong sequence. And that's coordinate control and combinatorial control. If you read over this, it should help. It's a little thick, but please come to my office hours if you have any questions about it. All right, and we're just going over it quickly just to kind of give you an idea. Now, we can regulate gene regulation, guys, at the transcriptional level. That is, we can regulate it right there. Either we transcribe or we don't. So if the activator proteins are there, it goes. If the activator proteins are not there, it doesn't go. If repressor proteins are there, it stops it. If repressor proteins are not there, it goes. Okay, so we can regulate right there at transcription. We can also regulate uh, after transcription. So I've transcribed the mRNA. Now what? I can actually inhibit it from attaching to a ribosome, and I can alter it. So this is RNA processing, and this would happen after transcription takes place. It just shows you how, more or less, remember that mammals like us, we don't have a lot of genes. However, we are able to make many, many different proteins from one gene. All right, and that just it's all based upon how that mRNA is spliced right here. So there's the primary transcript right there. Okay, and it shows that I can put these exons together 1, 2, 3, and 5, or I can put 1, 2, 4, and 5 together, and I can treat 3 as an intron. So here 3 is treated as an exon for this guy, and here 4 is treated as an exon and an intron for this guy. Okay, so that is why you and I can have the exact same DNA, exact same genetic code, but the protein later makes is slightly different from the protein one of you fine people make. All right, and it's not necessarily bad. It's just variation. That's how you get variation in a population. Could be slightly different skin color, slightly different eye color, slightly different height, whatever. And it's just because in our DNA, it's what you treat as exons and what I treat as introns and how I splice that together. This is also how your immune system cells actually kind of are very creative and come up with a whole bunch of different antibodies and so on and so forth. It's by splicing together different sections of DNA. Okay. Now, this is called mRNA degradation. Remember, you make the mRNA, and that mRNA is going to be read at the ribosome, where tRNA is going to bring the amino acids, but it's not going to live forever because it's living in a very hostile environment. And that hostile environment is going to begin to degrade and break down that mRNA. And as it degrades, once it's gone, it's done. Okay, it has to be transcribed from the DNA. So it says in bacteria, mRNA only lives for a very short time because it doesn't have a cap and tail. In eukaryotes, mRNA molecules can last as long as a few weeks. I've heard that. Some maybe say a little bit less, but okay. Therefore, they're continuously converted into protein. The nucleotide sequence found in the 3' cap area really affects how long the mRNA will live for, how long that cap is, basically. You don't really need to memorize anything here. I'm just trying to give you some other ideas of how we go from transcription to translation and what can interfere with it. That's what we're trying to do. As far as initiation of translation, I can inhibit that, okay? Because the body can actually receive signals or make regulatory proteins that can block the initiation of translation. It can block that ribosome from attaching to that mRNA. And so I put it right here for you, okay? It could be just because it was made but not quite ready to be read at all in the egg cell. The mRNA is already there, ready to rock and roll, but if there's no sperm, it's not ready to read those proteins just yet. It doesn't have the ability to do that. Another method, and this is in the egg cell cytoplasm, is that when the mRNA do not have very long poly A tails, when the gene is ready to be expressed, an enzyme adds a poly A tail to it so that it's going to last for a long, long time. Once again, in egg cells, the cell cytoplasm is loaded with mRNA. It's very heterogeneous, ready to be expressed. During the process of fertilization, there's a burst of translation since the process releases many translation initiation factors. Okay, all at once the mRNAs are expressed. Okay, protein processing and regulation. So what if I transcribe the mRNA, I even translate the mRNA? Can I regulate after I translate? Absolutely. I can modify the protein. So one thing we do is we modify proteins. So we make the protein and then other organelles such as the Golgi, that's a big one. It might attach like sugars to it. In the case of like a glycoprotein, so does the smooth endoplasmic reticulum. Those are heavy organelles to modify made proteins. And then some proteins are made, and then after they've been used, it's time to destroy them. So think of cyclin. Cyclin does not last in the cell cytoplasm the entire time. It's marked for destruction. And this is something you should know. right here. So some proteins are marked for destruction. Cells attach another protein called ubiquitin. Now, if you get tagged with ubiquitin, that means you're going to get chewed up by the machinery of the cell. So proteins that have ubiquitin marker on them are quickly destroyed by protein destroying enzymes called proteasomes. So the proteasomes are just these this basically the machinery, and it takes a protein and it destroys it. And what that means is it cuts it up into all pieces, parts. It's amino acid, and those things are usually recycled because the cell is the ultimate recycler of the cell. But that's how cyclin concentration, if you remember cyclin concentration, it goes up and down and up and down. Well, that's when it goes down, when it gets tagged with ubiquitin. So I just kind of put that right there for you as well. Okay, here's another situation of regulation. And this is where small non-coding RNAs, they're going to interfere with the mRNA and more or less attach to it and stop it. So you can see right here what they're going to do. This is called a microRNA right here. It's single-stranded. And this red and yellow guy right here is the mRNA. And it hydrogen bonds, because these are hydrogen bonds, it hydrogen bonds to the mRNA, and it can do one of two things. It is going to either hydrogen bond with it, and then basically it's with a protein right there. That purple thing's a protein, and it's going to degrade it, so now it's useless. It could do that before it was even red. It can even do it after, when it doesn't need to be red anymore. Or it can bond with it, and it can prevent this Big Mac, the ribosome, from attaching. And if the ribosome can't attach, well, lo and behold, I can't make a protein. Okay, so these guys are made by DNA. The code is in DNA to make these micro and small interfering RNAs. So miRNA in this situation stands for micro interfering RNAs. And then there's also siRNAs, small interfering RNAs. All right, but they basically do the same thing. They're just small interfering RNAs. And what they do is they stop after transcription or prevent translation. So I've got it right here for you to read over, but non-coding RNAs play multiple roles in controlling gene expression. This is some heavy stuff, but stuff that you just want to be aware of, and that's really about it. We know that 1.5% of the DNA is coded for proteins. That's it. Another small percentage of DNA is coded for rRNA, tRNA. It has also been found that a significant amount of DNA codes for small RNAs and non-protein RNAs, npRNAs. Okay, so there's small RNAs, smRNAs, and NP and ncRNAs and all this stuff. Many of these small RNAs and non-protein coding RNAs most likely serve a role as gene regulators. That's the big idea. That's what this chapter is about. Okay, effects on mRNA by micros and microRNAs, miRNA, and small interfering RNA, siRNA. Okay, so they're formed from longer RNA precursors that folds back on themselves, forming these double-stranded hairpin structures. You can see that right there. And then they break apart into a single strand. Okay, a dicer enzyme is going to come and do that. Some really nutso stuff. Okay, now one of the two strands is degraded, while the non-degraded strand forms a complex with a protein. That's that purple thing you see right there. Okay. The complex now binds with, most importantly, an mRNA, which shares the complementary base pair with. This either blocks translation or it degrades the mRNA, just like it's being shown right here. Most important. Remember, guys, what we've done all year, I always say, look at the figures. If you know what's going on in the figure, you're golden. It's just stopping it. That's all it's doing, okay? This could be like me right here, and I'm about to go get a Big Mac, and that's my wife right there, and she's like, heck no, and guess what? I don't get the Big Mac. That's as tough as it gets, really and truly. You could write children's book about this stuff, okay? There's also chromatin remodeling and silencing of transcription by small RNAs. They could, basically wind up that DNA real tight so it's not red. Stuff that we're still learning about. And I know some of you will have a lot of questions about it, but honestly, it's just not the time at this point. This is one of the last things we have to do. A program of differential gene expression leads to the different cell types in a multicellular organism. So remember, you started out as one cell, and then you were 2 cells and 4 cells and 8 cells and 12 cells, 16 cells and 32 cells. And those cells begin to differentiate and become specialized. So the basic program there is going to be, everyone's going to be the same type of cell. And then cells are determined Determined is kind of where they begin to specialize, and when they're fully specialized, they become differentiated. Remember, you're made of 100 trillion cells, and there's about 215 different types of those cells. That doesn't sound like a lot, but even within those classes, it gets a little bit more differentiated, especially the immune system cells. Trust me, they're amazing, okay? So, how does differentiation occur? And it's all based upon transcription factors that set the cell along its way. Okay, so it all starts with the mommy cell, the mommy egg. Okay, and here's what's going on. This is the egg cell. The egg cell is very, very, very special. Okay, the cytoplasm of the egg cell is very heterogeneous, and what that means is that it doesn't have all the same proteins in all these different locations. And then it gets fertilized by the sperm. Now it's more or less activated. Now these proteins right here, guys, these are called cytoplasmic determinants. And what they help the cell to do is begin the pathway of differentiation. So now that one cell, that zygote cell, is going to split. And now it's two. And as you can see right here, based upon what cytoplasm the cell got when it split, it's going to have different cytoplasmic determinants. And the green triangles and the orange spheres, what these cytoplasmic determinants serve as is, you guessed it, transcription factors. They will turn on unique sections of the DNA, and that will set the tone to begin the path of differentiation. Over here, this is where we're seeing a type of short signaling. This would be paracrine signaling right here, but it just shows within the embryo, now that these cells have become more differentiated, they actually become some signaling type cells, and they begin to signal nearby cells with paracrine signaling, as you can see the receptor, and that's going to help to also set the path for differentiation. So differentiation, it goes determination and then differentiation, and that determination right there is set forth by the fact that mom's egg cell already had in it, some proteins that were kind of different. Okay, so cytoplasmic determinants, I just put it right here, the particular specific genes expressed by a cell really help determine what types of cell it's going to become. So what tells the cell in the developing embryo what genes it will express? Before the egg was fertilized, it already had the stage pretty much set. The egg cytoplasm is not homogeneous, rather in certain areas it has proteins in it that are already coded by DNA, and it has the mRNA already be translated. Sometimes these highly influential proteins and mRNA in the egg cytoplasm are called determinants, meaning that the proteins already in the cell determine which genes will be expressed. They are transcription factors. And it just goes on to discuss that for you. Okay. This is called the sequential regulation of gene expression. So the step-by-step method of getting down to it. So this is how proteins like myosin are expressed, okay, in muscle cell. And this is a cytoplasmic determinant right here. It's called MyoD, okay? So it is a master regulatory gene. It's kind of like the P53 gene, all right? So MyoD, master regulatory gene. Right now it's off. When it's turned on, it's going to code for a protein called, you guessed it, MyoD. Okay, and it's going to be a transcription factor. So when MyoD is produced, you can see it right here, it serves as a transcription factor for other genes. So here's where the sequence is coming, and then that gene that it turns on is going to produce another transcription factor. But look at this transcription factor here. This transcription factor is going to turn on multiple genes. It's going to turn on a gene that codes for myosin, and it's going to turn on some other proteins that it says myosin, other muscle cell proteins, and cell cycle blocking proteins. Could be like tumor suppressor and stuff. Gets into some heavy stuff here. But that just shows you sequence right there. So once a cell differentiates, it it is determined and will follow that path indefinitely, even if the cell were to be placed into a different location within the developing embryo, continue on that path. So many regulatory genes control the differentiation process to help a cell complete its differentiation process. One such regulatory gene is MyoD. The gene codes for a protein also called MyoD. This protein is a transcription factor that binds to control elements in the enhancer region. All the genes that MyoD activates obviously have the correct control element combination to receive the signal. When MyoD activates these genes, the cells are differentiated, basically into muscle cells. Okay, so MyoD is a good example of a specific transcription factor, and genes that have the correct combo of enhancers will be turned on by it. Last thing, guys, are homeotic genes. So homeotic genes are genes that more or less code for body plan. So when you were developing, portions of the cells, became head cells and arm cells and leg cells. And they've studied a lot of fruit flies with this. And one of the neatest genes that they studied was called the bicoid gene. And the bicoid gene is the gene that, when it's activated, that becomes the front end of the anterior region, the head region, if you will, of the fruit fly. So you can see here's my developing egg. And here's the bicoid mRNA all fired up, ready to rock and roll. And this area ultimately will become the head of the developing fruit fly. Okay, so scientists, of course, got to mess with it, and what they did was they turned the bicoid off to see what would happen. So think about that for a second. I turned the bicoid off. Well, the bicoid determines the front end. So if I turn off the bicoid, that means that fly was actually born with two posterior, or two butts, if you will. All right, and it didn't live, but that's basically how that fly was born. So we just more or less, in this section of the chapter, we went through a lot of the fact, more ideas of what kind of regulates transcription and translation in eukaryotic cells. And we talked more or less about how I can regulate it at the transcriptional level, that is blocking transcription, not the right transcription factors there. After transcription, okay, such as RNA processing, okay, or degrading it from enzymes, or I can block translation. I can regulate the protein by making the protein. I can modify it, or I can tag it for destruction. Okay, I can interfere with the mRNA as well, meaning mRNA is made, I can tear it up. I can block it from being read. Okay, and then we also kind of got into the beginning, if you will, of how differentiation of cells takes place. First, determination by those cytoplasmic determinants, which act as transcription factors, turning on certain genes within those cells, and therefore that cell sets the stage to differentiate. Okay, and just kind of talking about that a little bit, and looking at something like a regulatory gene that more or less, once this guy turns on, that means it's going to set the stage for these cells to become muscle cells. So once MyoD's on, that's a muscle cell, pretty much. Okay, and then also some homeotic genes that control body plan. Pretty heavy stuff. All right, I think it's interesting, but certainly some of the more abstract things we've had to do all year, and always save. That's why I saved chapter 18 until now. Usually we get it done by early April. Okay, so from now on, we're going to finish some work this week on this. And then we're going to be also reviewing a bit this week. You guys are going to get your first FRQs this week. All right.